Hello, I'm back and I'm here to do Biology 1.4. So this whole chapter is about membrane transport. So a quick little side note before we begin. So basically a solution has two aspects. It has a solute and a solvent. So the solute is the substance that is dissolved in the other substance. Meanwhile, the solvent is a substance that dissolves the solute. So that would usually be water. That's just some context for the rest of this chapter. So the first thing we'll be talking about is what diffusion is. So diffusion is the movement of molecules from high to low concentration. High to low concentration is also called down the concentration gradient. That's like another term that is usually used. Diffusion in general is just due to random movements of molecules. So over time, the molecules will move around and things will be transported. Yay. Okay, so what affects the rate of diffusion? There are three things that we talk about in the book. There's temperature, concentration, and molecule size. So this is because certain sizes will not be able to use certain types of transport. Some will have to use active transport, some will have to use passive transport. We'll talk about that soon. It's also important to note that cell membranes are semi-permeable, which means that only certain molecules can pass freely. So as you can see in this drawing, the bigger molecules bounce off the walls, meanwhile the small ones are able to go in and out freely. This means that it is semi-permeable. So simple diffusion across the membrane, so it goes from high to low concentration, and as you can see, this is a very simple form of diffusion, simple form of transport. It is passive, meaning that no energy is required. You can think about this as in like paying for a bus ticket or something, that in this way, the molecules are basically, or substances, are just walking across. They don't have to pay for a bus ticket. No energy is required. Meanwhile, if it's active transport, you do need to pay, aka you need to add ATP, which is a form of energy. But no, this one is passive. This is usually used for small substances, non-ionic substances, and for fat-soluble molecules. So generally speaking, very small things. An example of this is oxygen. Oxygen can just go back and forth with simple diffusion. Well, not back and forth. They can go from high to low concentrations because it only goes one way. Now the next one is facilitated diffusion, which also goes from high to low concentrations. It is also passive, and it is used for ionic compounds and fructose. Essentially, what you need is a channel protein, which is filled with water, where the molecule or compound, fructose, whatever, can move down. It can just go through it. Yeah, exactly. I don't know what I'm saying right now, but I think you all get it. And I drew a molecule, a different one, or a different substance that was a bit bigger and like more like triangle-y. And I wrote that it doesn't fit because essentially, as I said, it's semi-permeable, which means that some substances can move freely, others can't. So now we're going on to osmosis. So the osmosis is the diffusion of water across a semi-permeable membrane. It also goes from a low solute to a high solute concentration. No ATP is needed here either, so no energy is needed. So essentially, it goes with time, and instead of moving, as we said, instead of moving the actual like molecules or compounds, you're moving the water. The water is moving from one to the other side. So if you want to look at how different types of concentrations will affect an animal cell and plant cell, we'll go through that now. So the first type is hypotonic. This means that the hypotonic solution is less concentrated than the cell. What happens for an animal cell is that all of the water goes into the cell and it leads to something called lysis. So the cell basically explodes because all the water is going into it. Then for a plant cell, it leads to the cell being turgid, aka really stiff and hard and like, you know, like really, really filled. That's the only way I can explain it. Like turgid is like, it's really stiff and like hard and not good. Okay, not good at all. Then there is an isotonic solution, which is basically, it has the same concentration as in the cell. This, for an animal cell, just leads to the normal state of the cell. It's fine. There's H2O going in, H2O going out. It's equal, it's good. It's all fun and fresh. Now, for the plant cell, it leads to it being flaccid, which, to be honest, is normal. If that sounds negative, but it's an isotonic solution, so your plant cell is perfectly fine. Then there is a hypertonic solution, which essentially when there's a higher concentration than in the cell. This is pretty good to think that like hyper is usually seen as like, you know, a peak. So you can think about it in that way, so you remember that it's higher. So for an animal cell, it gets crenated. This is with an N, not cremated. Essentially all of the H2O leaves, it gets dried out. And then for a plant cell, it leads to plasmolysis, which is the same thing, except remember that it has a cell wall, so the cell wall still stays. So let's talk about estimating osmolarity. So osmolarity is a measure of solute concentration, which refers to the number of particles per liter in the solution. So the unit is OSM per liter. So osm per liter. I highly doubt that's how you say that, but I'm going to pretend that that is how that is said. Yes. So when you're estimating it in plant tissues, you can think hypotonic solution, the tissue will gain water, it will become stiff and increase in mass. In isotonic solution, there will be no change, and in a hypertonic solution, there will be water loss, so loss in mass, and it will become limp and soft. So usually we did this in class with like a potato. We had like potato strips and we tested them. So when it comes to organ and tissue transplants, you must keep the organ or tissue in isotonic solutions, which is around 0.9%, because otherwise it won't stay alive. So if you put it in a hypertonic solution, it will have lysis, and if you put it in a hypotonic solution, it will be, I'm sorry, I mixed it up. I meant crenated, and if you put it in a hypotonic solution, it will have lysis. So far, we've only spoken about passive transport, but now let's talk about active transport using carrier proteins. So you have these protein pumps, and you attach ATP to them, and then over time, the protein pump will change shape. But only if energy is added, otherwise it won't. And this one actually goes from low to high concentration, so this is up the concentration gradient. So you go from low to high, and it is very selective, so like only certain substances can go through it. And an example is the sodium-potassium pumps in the nerve cells. Yay! Okay, so next thing is exocytosis. This is the removal of substances from a cell using vesicles. So essentially, you have a cell, and the material to be transported is enclosed inside a vesicle within the cell. Then the vesicle carries the material to the plasma membrane, and the vesicle fuses with the membrane, and the material is released, which is called secretion. Yeah, that's a pretty simple process, actually. So now let's talk about bulk transport, which is also called vesicular transport. So essentially this is active transport, so you need ATP. So you have a white blood cell and you have bacteria. A pit forms in the membrane of the white blood cell, and the membrane encloses the particles. A vesicle is formed, and then the vesicle moves into the cytoplasm. Lysosomes release enzymes into the vesicles, which are phagosomes, and the bacteria is digested. So endocytosis is the intake of large particles by engulfing them. So there is phagocytosis and penocytosis. So phagocytosis is the intake of solids, meanwhile penocytosis is the intake of solutions, usually by the intestinal cells. So endo is the intake and exo is the removal, if you remember from like 2 steps before. So the last part of this chapter is protein transport and secretion. So this is how vesicles move materials within cells. So next to the nucleus, the ribosomes on the rough endoplasmic reticulum produce proteins. Then vesicular transport is used to transport the protein to the Golgi apparatus. At the Golgi apparatus, the protein is modified and repacked. Then the Golgi pinches off vesicles for transport towards the membrane for secretion. But there are 2 options here. Either it goes to the cell membrane for secretion, or the Golgi pinches it off and lysosomes with digestive enzymes are created. So those are the 2 options there, and that's how that works. So thank you so much for watching. I hope you subscribe, like, and comment on this video if you want to. You can follow me at Johanna Frennert on Instagram if you feel like it, and I hope you learned something. Goodbye!